I'm Carly Miller at the University of Illinois, and today we're discussing invertebrate diversity, or the different invertebrate phyla. So remember, an invertebrate is an organism that is lacking a backbone. And later we will contrast that to the subphylum Vertebrata, which definitely has a backbone here. So we're going to go through step by step the different phyla of invertebrates, and what we want to identify is specific features that you can tie back to a specific phylum. And so we're going to go through symmetry, the modes of life, the feeding modes, as well as any particularly special characteristics that particular phylum exhibits. So we're going to start with a very basic organism in the phylum Porifera. So you may recognize Porifera as sponges. So sponges, again, these are all going to be, or most of them will be marine organisms. And so we're going to start with sponges. So again, these organisms are asymmetrical. Some of them can be radial. Their mode of life is that they're encrusters, meaning they grow on surfaces. They grow on other organisms sometimes even. They're epifaunal, meaning they're on the seafloor, not within the seafloor. And they're sessile. So I don't know if you've ever seen a sponge move, but I have not. And so that means that they're sessile. Their feeding mode, because they're sessile, they have to be filter feeders, more or less. So Porifera, the phylum Porifera, they are all filter feeders. And there's one exception that is carnivorous. The cellular structure of the phylum Porifera is that they don't really have any true tissues by definition. So they are composed of cells, but not a tissue, which is a very specific organization of those cells. Lastly, a piece of great information is that a group of sponges is called a sleaze. I've never heard that before, but pretty interesting. The next phylum we'd like to consider is the phylum Cnidaria. So this is composed of corals, anemones, and jellyfish. So if you're thinking about a jellyfish that you may have seen, or a coral that you've snorkeled over, what you might notice is that they are radially symmetrical. The mode of life is epifaunal and sessile. So again, corals are going to grow on the seafloor, and they're not going to move anywhere. So there are a few vagrant there are a few vagrant cnidarians, but most of them are pelagic and planktonic. So remember, that means they're kind of floating with the currents in the ocean. So a jellyfish, it's radially symmetrical. It's not going in a particular direction, but it does move. So the phylum Cnidaria, they are composed of carnivores. So they are vagrant, and they actually are passive carnivores, meaning they can't hunt in a particular direction or a particular food source, but they do eat live prey. And how they do that is they use stinging cells called cnidoblasts. And those are usually activated mechanically or by touch. So that's why, other than preserving corals, you're not supposed to touch them, is that they actually can sting you. And I'm sure you've heard of jellyfish actually being quite dangerous or deadly to humans because of these cnidoblast cells. And so they're able to stun their prey and then ingest them. Corals have stony skeletons, and so they are well preserved in the fossil record. They have a thin layer of soft parts, but we have a great bulky skeleton that is then well preserved in the fossil record. Additionally, organisms in the phylum Cnidaria can be solitary, or they can be colonial organisms. So the functional unit of a colony is just one individual organism, but each organism is then either genetically identical or very similar to its neighbor. And so those are put together in an assemblage called a colony. Moving on to a much less dangerous phylum, this is the phylum Bryozoa. They're similar to a sponge, maybe in structure, but they're definitely within their own group. And so these guys are mostly asymmetrical. They're benthic, epifaunal, and sessile, so they're not going to be moving. And we do have a couple of great fossils of different parts of the bryozoa, not necessarily a full, complete fossil of a bryozoan, because they're rather delicate, actually. So here you can see a stem, if you will, or one of the one of the more robust parts of a bryozoan organism. Additionally, because we've mentioned that the bryozoans are epifaunal and sessile, again, meaning they're not going to move along that sea floor, they are again going to be filter feeders. The phylum Bryozoa, they have a specific feature called the lophophore. And they use this lophophore to filter feed, and that's just a specific structure that specifically the bryozoans use. The next phylum we'd like to consider is Brachiopoda. Again, these are your brachiopods, and they are widely distributed in the fossil record. They're well preserved often, but there are several different shapes of brachiopods, and I'll show you some examples so that you can identify them. A couple of the main features of this phylum is that their feeding mode is that they're filter feeders, because again, we've identified that they're benthic, epifaunal, and sessile, meaning they're not moving. And these particular organisms are not colonial. Brachiopods are similar to bryozoans in that they also use a lophophore. So this particular structure is specific to the phylum Brachiopoda and the phylum Bryozoa. Brachiopods attach to the seafloor using a pedicle, which is a fleshy appendage that comes out the back of the shell and sticks to the seafloor, and they can use that pedicle for attachment. Brachiopods also have a calcium carbonate exoskeleton. So their shells are composed of calcium carbonate. And what we want to note about the phylum Brachiopoda is that they're one of the most successful organisms in the Paleozoic era. All right, lastly, we want to consider the symmetry of brachiopods because it's specific to this phylum. So the symmetry of these organisms is that it's bilateral symmetry, but it's perpendicular to the plane of commissure. The plane of commissure is the surface or the plane where two shells come together. For example, on this bivalve, you can see that there's a top shell and there's a bottom shell, and there's a plane that separates the two shells. And that plane is called the plane of commissure. So with brachiopods, they too have a top shell and a bottom shell and a plane of commissure, but their symmetry is such that it's not the same on the top and the bottom, but it's actually on the same from right to left. So we call that perpendicular to this plane. So moving forward, the next phylum we'd like to consider is phylum Mollusca. And so this is a diverse phyla of shelled organisms or formerly shelled organisms. And we've divided this phylum into many classes, but we'd like to focus in on three particular classes: the class Bivalvia, the class Gastropoda, and the class Cephalopoda. So the first class in phylum Mollusca that we'd like to consider is the class Bivalvia. So these are clams, mussels, oysters, and scallops, for example. Their symmetry is bilateral symmetry, but it's parallel to the plane of commissure. So this is, again, in contrast to the brachiopods, which had symmetry perpendicular to that plane. So just to demonstrate that symmetry, we have a top shell and a bottom shell, and we have our plane of commissure, which is that plane where those two shells meet. And again, you can see here on this bivalve that the top and the bottom are mirror images of each other. And so the symmetry is along that plane, and that's parallel to the plane of commissure. The mode of life of bivalves is epifaunal, but they can also be infaunal, meaning they have the capability to bury themselves or survive on the seafloor. The feeding mode of the class Bivalvia is filter feeders, and they use a siphon to filter the seawater. And it's similar to the structure of a lophophore found in bryozoans and brachiopods, but a little bit different, and is called a siphon, and is specific to the class Bivalvia. And just one note here, so we talked about the symmetry being bilateral in general, and parallel to the plane of commissure in this class, but oysters and scallops in particular are generally asymmetrical, actually, so we want to look out for that. The second class that we'd like to consider in the phylum Mollusca is the class Gastropoda, or your gastropods. So these are snails, slugs, and they can be very diverse as far as the level of species goes. The symmetry of gastropods is that they're asymmetrical shells, and they have bilaterally symmetrical bodies, actually. The mode of life of the class Gastropoda is that they're epifaunal, so they live on the seafloor, and they're vagrant, so they move. So if you think of a snail or a slug, you know that those are moving around. And you can think of a slug as an example of a bilaterally symmetrical body. So that's a good example. You could picture that in your mind, maybe what a slug looks like, splitting it in the right and left halves. And a snail is basically a slug with a shell. And so you can picture how the shell is maybe asymmetrical, but the organism that lives it, or the soft parts, are bilaterally symmetrical. And the feeding mode is interesting with the class Gastropoda because all feeding modes are represented, including carnivores. So look out for those slugs. The third class in the phylum Mollusca is class Cephalopoda, or your cephalopods. This includes squid, octopi, ammonoids, and nautiloids, and many can be extremely large. The symmetry of cephalopods is that they're bilaterally symmetrical with their bodies and their shells. Their mode of life, if you can think of a squid, it's nectonic, which meaning it's moving in a particular direction in the water column. If you think of octopi, they're actually going to be benthic, but obviously vagrant. They do move around. And they are active predators. So stay away, observe closely, but stay away. So if they're active predators, then their feeding mode is that they are carnivorous. Some cephalopods exhibit a single piece of a coiled shell, such as this nautilus, and they have a siphuncle in them, a siphuncle, which is a tube that brings in water to the shell and controls the buoyancy of it in the water column, meaning it controls the position of the organism in the water column. And an interesting note about cephalopods is that they generally have above average intelligence. So again, these are invertebrates, they're mollusks, and they are highly intelligent. And this is correlated to actually very good eyesight. And they're very fast movers. So I would beware of the class Cephalopoda. All right, moving to another phylum of invertebrates, we want to consider arthropods. So the phylum Arthropoda. These are insects, trilobites, which are extinct, crabs, barnacles, spiders, scorpions, and lobsters. Add a little butter there, right? Arthropods are the most successful and diverse and numerous organisms in one phylum. So it's something to consider. You want to note that. This is a very diverse phylum. Their symmetry is bilateral, though. So that's a specific symmetry. So even though this phylum is very diverse, you have a lot of radiation there, a high number of organisms included in this phylum, they all are bilaterally symmetrical. So we can gather some information from that symmetry again, but the mode of life in the phylum Arthropoda is they can basically be anything. And if you were paying attention to the list of examples, insects, trilobites, crabs, lobsters, scorpions, you'll see that they're representing marine organisms, freshwater organisms, and terrestrial organisms. So a very diverse group here. Their feeding mode, again, across the board. Something you want to consider if you've identified an organism as an arthropod is that you need to pay very close attention to the shape, the structure, the functional morphology of that organism so that you can start to hypothesize what this organism ate and how it lived. So one of the key characteristics of an arthropod is that it has a chitinous exoskeleton, so a skeleton on the outside of the organism, and it's divided into segments, which is usually where humans get a little weirded out. So here we have an example of an arthropod, and this is a trilobite. Again, these are extinct, but you can clearly see it has segments where each of those body parts are almost identical, if not identical, to the one before it. So it's a segmented organism, and that's specific to arthropods. And again, this trilobite fossil is actually a great one because it also exhibits the bilateral symmetry that the phylum exhibits. The last phylum we'd like to consider of invertebrates is called Echinodermata. So this is represented by sea stars, brittle stars, sea urchins, sea cucumbers, sand dollars, echinoids, and crinoids. So their symmetry is pentameral symmetry with some exceptions, and they have a calcium carbonate endoskeleton, meaning inside their bodies. So they're covered by a soft layer of tissue on the outside. There's two classes we'd like to consider within the phylum Echinodermata, Crinoidea and Echinoidea. The first class is Crinoidea, and this is represented by sea lilies and crinoids, hence the name. Living crinoids are generally unstalked, meaning they don't have a long stem, a segmented stem coming up to almost kind of a like a flowery shape. I think these are like sea lilies, basically. But so living crinoids are generally unstalked, but there have been reports in deep water that there are some stalked crinoids still living today, which is unusual. Extinct crinoids were almost by rule, all stalked. So if you had a chance to play in your playground as a child and you had a gravel bottom to your playground, you might have found something like this, which looks like a segmented little peg, if you will. And I used to find these in my local playground, and I never knew what they were. And here I am, a geologist, and I find out that these are crinoid stems, actually. And so they could be several feet in length, and they actually have easily observable segments. And something you want to be aware of when you're considering a crinoid stock is that upon first glance, they actually look radially symmetrical, right? They're almost a perfect column, or a cylinder. But if you look closer, you actually see that in the middle of the column there's a star, or it looks like a star. And so if you were to count the points on the star, there's 5. And so that actually then knocks it into the pentamerally symmetrical structure. So crinoids are benthic, epifaunal organisms, and they're sessile. So if you can think about having a stalk, that's not the same as a leg, right? It's more like a stem. And so that organism is going to be rooted to the seafloor. So it's going to be benthic and epifaunal and sessile. And because they're sessile, they're again going to be filter feeders. The second class in Echinodermata is Echinoidea, and there's regular echinoids and irregular echinoids. Regular echinoids are sea urchins, and they're epifaunal, they're vagrant herbivores. They scrape algae off the rocks with something called an Aristotle's lantern, which we'll look at in closer detail. And regular echinoids have long spines, actually, so that's a key feature of those particular sea urchins. An irregular echinoid is actually what you might call a sand dollar. So if you think about a sand dollar, they're actually rather flat. They're smooth, generally in structure, and they've actually been reduced to having tiny spines as opposed to long spines with the regular echinoids. So these irregular echinoids have greatly reduced spines. They're still vagrant, but since they're smoother, they can actually become infaunal, and they can bury themselves in the ground. So we have regular echinoids, which are epifaunal, and we have irregular echinoids, which are smoother, and they're infaunal. Additionally, irregular echinoids are detrital feeders, meaning they mix up the sediment and they actually eat the vitamins and the nutrients that are buried within the sediment. And the regular echinoids are herbivores. So here we've considered a diverse group of invertebrates. We've gotten very specific with the characteristics that are exhibited within each phylum. The important thing about what we've discussed is that you can deduce most of that information from looking at a specimen that you find. And so we can deduce a lot of information from the symmetry, the shape of the organism, and those are very specific. And so if you can identify those features, you can actually place this organism taxonomically into a very particular phylum.